The atoms in your body right now are between 13.8 billion and 4.6 billion years old. The iron in your blood was forged in the core of a dying star that collapsed before our solar system existed. The calcium in your bones was synthesized in the nuclear burning shells of a red giant during its final stages of fusion. The carbon forming the backbone of every protein, every strand of DNA, every cell membrane in your body passed through the interior of at least one stellar core before arriving here, in this configuration, reading these words. You are not made of the Earth. You are made of stars that preceded the Earth by billions of years. And the moment you die, the precise moment that the biological organization maintaining this particular arrangement of atoms ceases, those atoms do not go anywhere. They do not disappear. They do not become something lesser. What happens to them is, from a physicist's perspective, one of the most extraordinary transitions in nature. The controlled, organized, precisely maintained order that constitutes a living body gives way to an entirely different kind of order, following entirely different rules, and the atoms that constituted you begin a journey that will carry them, over the next several billion years, through forms and configurations that a physicist can trace with more precision than most people would find comfortable. Let me start with what stops. The living body is a thermodynamic engine running at a permanent distance from equilibrium. Every cell in your body is continuously consuming free energy available to do work, and using it to maintain structural and chemical order that would not exist without that continuous energy input. This is what life is in the physicist's description. A local decrease in entropy maintained by a continuous throughput of energy. The cell maintains protein structures that would spontaneously unfold in the absence of active maintenance. It maintains electrochemical gradients across membranes that would dissipate in seconds without the ion pumps constantly expending ATP to rebuild them. It maintains the spatial organization of organelles, the specific composition of its lipid bilayers, the precise topology of its chromatin. All of this requires energy. All of this is running, every second of every day, in every one of your approximately 37 trillion cells as long as you live. The energy source is ultimately food, reduced carbon compounds that the body oxidizes, extracting the free energy difference between the reduced state of the fuel and the oxidized state of the products, capturing that energy in the phosphate bonds of ADP, and using ADP to power everything from ion pumps to motor proteins to the synthesis of new molecules. The rate of this energy throughput, the metabolic rate, determines how far from thermodynamic equilibrium the body sits at any given moment. A resting adult human dissipates approximately 80 watts of power as heat, about the same as an incandescent light bulb. During intense exercise this rises to 700 watts or more. This power output represents the continuous expenditure required simply to maintain the organized state that constitutes a living body. The moment the heart stops and circulation ceases, the energy supply to the cells is cut. This is the beginning of what pathologists call autolysis and what physicists would describe as the thermodynamic relaxation of the system toward equilibrium. It does not happen all at once. It happens in a sequence, beginning with the most energetically expensive and rapidly degrading processes and progressing systematically toward the most stable. The first structures to fail are the ion gradients across cell membranes. The sodium-potassium ATPase pump, the enzyme responsible for maintaining the steep concentration gradient of sodium outside and potassium inside every cell, which is the basis of electrical signaling in neurons and muscle cells, runs on ADP. Without blood flow delivering oxygen and glucose, ADP synthesis in the mitochondria stops within seconds to minutes. Existing ADP reserves are depleted within minutes. The sodium-potassium pump stops. Sodium floods into the cell along its concentration gradient. Water follows by osmosis and the cell swells. Potassium leaks out. The membrane potential, the electrical charge difference across the cell membrane, normally around -70 millivolts in a neuron, collapses. Every electrical signal that was your consciousness, your sensation, your identity ceases. Not because the neurons are destroyed. Because the electrochemical gradient that generates their signaling is gone. The hardware is still there. The power is off. Within minutes to hours, the lysosomes, membrane-bound organelles within each cell that contain digestive enzymes including proteases, lipases and nucleases, begin to rupture. The lysosomal membrane, like all cell membranes, requires energy-dependent processes to maintain its integrity. As the cell's ADP is depleted and the membrane potential collapses, the lysosomal membrane fails. The digestive enzymes, cathepsins, phospholipases, nucleases, are released into the cytoplasm and begin digesting the cell from within. This is autolysis in its most literal sense. The cell is eating itself. The protein complexes that maintain the exquisite organization of the cytoplasm are broken down into their component amino acids. The DNA in the nucleus, protected during life by histones and nuclear architecture, begins to be degraded by denases. The lipid bilayers of the cell membranes are cleaved by phospholipids. The cell, in the absence of the energy inputs that maintained it, is dismantled by the same enzymatic machinery that it used for routine recycling during life, except now there is no rebuilding, only breakdown. The breakdown products, amino acids, fatty acids, nucleotides, sugars, are small molecules that can diffuse through the increasingly permeable cell membranes. They enter the interstitial fluid between cells. And they are food. This is where the next layer of the story begins because the body does not decompose in a vacuum. The human body carries, within the gastrointestinal tract and on the skin surface, approximately 38 trillion bacteria, a number comparable to the total number of human cells. These bacteria are alive during your life, maintained in their particular ecological balance by the immune system, the gut mucosa, the antibacterial properties of various secretions, and the continuous competition between bacterial species for resources. The moment the immune system stops functioning, which happens within hours of death, as the neutrophils and macrophages that constituted the cellular immune defense lose their energy supply and cease their surveillance, the bacteria are free. The commensal bacteria of the gut, which were contained within the intestinal lumen by the intact gut wall and the immune system, begin to penetrate through the gut wall into the surrounding tissues. And they carry with them their metabolic machinery, their enzymes, their capacity to break down organic molecules and extract chemical energy from them, their ability to release gases as metabolic byproducts. The bacteria begin consuming the breakdown products of autolysis, the amino acids, fatty acids, and sugars that are diffusing out of dying and dead cells. They use anaerobic fermentation pathways to extract energy from these substrates, producing gases in the process. Hydrogen sulfide, H2S, is produced by the reduction of sulfur-containing amino acids like cysteine and methionine. Carbon dioxide, CO2, is produced by decarboxylation reactions. Methane, CH4, is produced by methanogenic archaea. Ammonia, NH3, is released by the deamination of amino acids. These gases accumulate in the soft tissues and body cavities, causing the bloating and discoloration characteristic of the early decomposition stage that forensic pathologists call putrefaction. The skin, which is relatively impermeable during life due to the continuous renewal of its outer layer, is now static, not being replaced, being digested. The green-black discoloration of putrefaction is caused by the reaction of hydrogen sulfide with hemoglobin to form sulfhemoglobin, and then with iron from hemoglobin to form iron sulfide, FeS, a black compound. The iron that was carrying oxygen through your circulatory system is now forming iron sulfide in your decaying skin. But the bacterial decomposition of the soft tissues is not the end of the atom's journey. It is the beginning. The products of bacterial decomposition, amino acids, fatty acids, sugars, and their further breakdown products, enter the soil. The bacteria themselves are consumed by other bacteria, by fungi, by protozoa, by nematodes. The organic matter that was your body enters the food web of the soil ecosystem, which processes something like 90 billion tons of organic matter per year globally and is, from a geochemical standpoint, the planetary recycling system for organic carbon and nitrogen. The amino acids in your proteins, each one containing a nitrogen atom in its amino group, release that nitrogen through a sequence of microbial processes. Amino-mineralizing bacteria break the amino acids down to ammonia. Nitrifying bacteria, Nitrosomonas and Nitrobacter, oxidize ammonia to nitrite and then to nitrate through the two-step nitrification process. The nitrate enters the soil water and is taken up by plant roots, incorporated into plant amino acids and nucleic acids, and eventually eaten by animals including humans who incorporate it into their own proteins. The nitrogen atoms that were in your body's proteins will, within decades, be inside the proteins of other living organisms. The carbon story is even more direct. The carbon dioxide produced by bacterial respiration and fermentation during your decomposition enters the atmosphere. In the atmosphere, carbon dioxide is the substrate for photosynthesis, the process by which plants, algae, and cyanobacteria use light energy to reduce CO2 to carbohydrate, fixing carbon from the atmosphere into organic molecules. A carbon atom that exits your body as CO2 can be fixed into a glucose molecule within days, if it happens to be absorbed by a leaf in the right conditions. From the glucose, it can be incorporated into cellulose, becoming the structural material of a tree, or into starch, becoming food energy for an animal, or into a protein, becoming part of the cellular machinery of a living organism, within a time frame of weeks to years. The average residence time of a carbon atom in the atmosphere is approximately 4 years. This does not mean your carbon atoms will cycle back into living organisms within 4 years. Some fraction of your carbon will enter long-lived carbon reservoirs like wood, peat, or deep soil organic matter where it can remain for decades to centuries to millennia before returning to the active carbon cycle. A small fraction of the carbon from any dead organism eventually ends up in sediment, where, over geological timescales of tens to hundreds of millions of years, it can undergo transformation. Organic carbon buried in anoxic sediments is heated and compressed by the weight of overlying sediment and eventually, at temperatures above approximately 150 degrees Celsius and pressures above approximately 200 megapascals, undergoes catagenesis, the thermal cracking of large organic molecules into smaller hydrocarbons, petroleum and natural gas. Your carbon might become petroleum. It has done this before. The carbon in petroleum existed in living organisms hundreds of millions of years ago. It is doing the same thing again. Now I want to think about the specific atoms themselves, not the carbon and nitrogen chemistry but the nuclear physics. Every atom in your body has a nuclear identity, a specific number of protons defining the element and a specific number of neutrons defining the isotope. Most of the atoms in your body are stable isotopes. They will remain the same element indefinitely. The hydrogen in your water is primarily hydrogen 1, a single proton, no neutrons, which is stable essentially forever. The carbon is primarily carbon-12, stable. The oxygen is primarily oxygen-16, stable. The nitrogen is primarily nitrogen-14, stable. These atoms will cycle through living organisms, through the atmosphere, through the ocean, through soil, through rock, for as long as the Earth exists. They were stable before the Earth formed and they will be stable after the Earth is gone. But a small fraction of your atoms are radioactive. Carbon-14, formed continuously in the upper atmosphere by the bombardment of nitrogen-14 by cosmic ray neutrons, makes up approximately 1 atom in a trillion of the carbon in all living organisms, maintained at a constant level during life because living organisms continuously exchange carbon with the environment. When you die, the exchange stops. The carbon-14 in your body begins to decay at a known rate, a half-life of 5,730 years, emitting a beta particle, an electron, as each carbon-14 nucleus converts to nitrogen-14. This is the basis of radiocarbon dating. The ratio of carbon-14 to carbon-12 in a dead organism's remains decreases at a known rate, allowing the time of death to be calculated with precision. Your radioactive carbon atoms are, in a very real sense, a built-in clock that continues running after you die, broadcasting the time of your death to any physicist with a mass spectrometer. Beyond carbon-14, your body contains trace quantities of other radioactive isotopes. Potassium-40, which has a half-life of 1.25 billion years and makes up 0.012% of all potassium. Rubidium-87, which makes up 28% of natural rubidium and has a half-life of 49 billion years. Uranium-238, present in the body in microgram quantities, with a half-life of 4.47 billion years. These long-lived radioactive isotopes in your body are decaying right now, emitting radiation. They were incorporated into your body from food and water, where they are present in trace amounts because they have been present in the Earth's crust since the Earth formed from the solar nebula 4.6 billion years ago. When you die, they continue decaying in your remains, in the soil your remains become, for timescales that dwarf the entire history of life on Earth. Here is the part of this story I find worth dwelling on. The physicist Erwin Schrodinger, who in 1926 formulated the wave equation that became the foundation of quantum mechanics, and who in 1944 wrote a small book called What is Life? That influenced Watson and Crick in their discovery of the structure of DNA, described life as the capacity to maintain order against the thermodynamic tendency toward disorder. He used the phrase feeding on negative entropy, or negentropic feeding, to describe the way organisms extract order from their environment by consuming high-entropy energy sources, food, and expelling higher-entropy products, heat and metabolic waste. What Schrodinger was describing is what physicists now formalize in the framework of non-equilibrium thermodynamics. The living organism is a dissipative structure, a term coined by Ilya Prigogine at the University Libre de Bruxelles, who won the Nobel Prize in Chemistry in 1977 for his theory of dissipative structures and the thermodynamics of systems far from equilibrium. A dissipative structure is a pattern maintained by the continuous flow of energy through it, like a candle flame, which maintains its characteristic shape and chemistry not by storing anything but by continuously consuming fuel and expelling combustion products. The flame is a process, not a thing. And so, in Prigoghine's framework, are you? The moment you die, the process stops. The dissipative structure dissolves. The atoms that constituted the pattern remain, exactly as they were at the moment of dissolution, and immediately begin following the thermodynamic gradients that the living process had been holding at bay. The sodium floods in. The lysosomes rupture. The bacteria cross the gut wall. The proteins unfold. The gradients dissipate. What was highly improbable, the specific arrangement of atoms that constituted you, becomes progressively more probable arrangements, moving through states of lower and lower free energy until the atoms reach configurations that can persist without continuous energy input. Stable inorganic compounds, soil minerals, atmospheric gases, ocean-dissolved matter. But here is what I want you to understand about those atoms. They are not diminished by this process. The iron atom that was in your hemoglobin carrying oxygen to your brain does not become less iron when it forms iron sulfide in your decomposing skin, or when the iron sulfide dissolves and the iron ion enters the soil water, or when it is taken up by a plant root and incorporated into a ferredoxin protein, or when that plant is eaten by an animal and the iron is incorporated into that animal's hemoglobin, carrying oxygen to that animal's brain. It is the same iron atom throughout. It has the same 26 protons in its nucleus, the same electron configuration, the same chemical properties. What changes is the organization it participates in, the molecular context, the functional role, the thermodynamic state. The atom itself is unchanged. The physicist Richard Feynman, in a lecture at the California Institute of Technology in 1961 that was later published in the Feynman Lectures on Physics, described the atom as follows. If, in some cataclysm, all of scientific knowledge were to be destroyed, and only one sentence passed on to the next generation of creatures, what statement would contain the most information in the fewest words? I believe it is the atomic hypothesis, that all things are made of atoms, little particles that move around in perpetual motion, attracting each other when they are a little distance apart, but repelling upon being squeezed into one another. What Feynman was pointing at is the extraordinary durability and identity of the atom as the fundamental unit of ordinary matter. The atoms in your body have been through stellar interiors, interstellar space, planetary accretion, geological transformation, ocean chemistry, and at least one other living organism before they got to you. They will go through all of these things again. Your lifetime is a brief episode in their much longer history. The nitrogen in your DNA right now was once in the atmosphere as N2, fixed into soil by rhizobium bacteria in root nodules of a legume plant, incorporated into a plant protein eaten by an animal, excreted as urea, returned to the soil as ammonia, nitrified to nitrate, taken up by another plant, eaten by the animal whose protein you ate, and then built into your DNA by your own cellular machinery. Before that, it was in the gas cloud that collapsed to form the solar system. Before that, it was synthesized in the core of a star by the CNO cycle, the carbon-nitrogen-oxygen nuclear fusion cycle that produces nitrogen-14 as a byproduct while converting hydrogen to helium. The nitrogen in your body was made in a star that no longer exists. Its nuclear identity has not changed since the moment it was synthesized. It has simply been through different configurations. After you die, it will be through different configurations again. Within decades, that nitrogen will be in the proteins of plants and other animals. Within millions of years, if it enters deep burial, it may be incorporated into rock as organic nitrogen, released during metamorphism, returned to the atmosphere. Within billions of years, when the sun expands into a red giant and the surface of the Earth is heated beyond the stability of its crust, those atoms will be returned to space, joining the interstellar medium from which new stars and planetary systems will form. Some of them will be incorporated into new planetary bodies. Some of those planetary bodies may develop conditions suitable for chemistry of the kind that, 4.6 billion years ago on this planet, gave rise to the first self-replicating molecules and eventually, through 3.8 billion years of evolution, to you. There is one more thing worth saying. The atoms in your body are indistinguishable from the atoms that are not in your body. The oxygen atom in the water you drink right now is physically identical to the oxygen atom in the air outside, which is physically identical to the oxygen atom in the rock under your feet, which is physically identical to the oxygen atom that was in a dinosaur 65 million years ago. The distinction between your atoms and not your atoms is a biological and organismal distinction, not a physical one. The atom does not know it is yours. What makes it yours, temporarily, contingently, for the duration of your life, is the pattern of organization that includes it, the molecular structures, the metabolic processes, the information encoded in the genome that directs the specific placement and use of each atom in the particular role it plays in maintaining the dissipative structure that is you. When the pattern ends, the atoms are released, not destroyed, not diminished, released, back into the common pool of matter that has been cycling through stars, planets, oceans, atmospheres, and living organisms since the first atoms were forged in the first few minutes after the Big Bang. You borrowed them. You organized them into something remarkable, something capable of asking what happens to atoms after death. And now you are returning them. They will be remarkable again. They have always been remarkable. Remarkableness is not something you added to them. It is something they are capable of, in the right configuration, under the right conditions. The conditions are temporary. The atoms are not.